Good afternoon. My name is Paul Hallenbeck. I'm the founder, president, and chief scientific officer of Seneca Therapeutics. I'm going to tell you a lot about Seneca Valley virus today, but before I do, I just want to tell you a really quick story about the serendipitous discovery of this particular virus called Seneca Valley virus. So in 2002, I was head of R&D at Novartis' cell and gene therapy enterprise when my team was testing an oncolytic adenovirus, and we were testing a variety of different cancer cell types as well as controls. And what we saw is very potent killing on these on these specific cancer cell types and no killing on the controls. This was pretty profound for an adenovirus, so it made us suspicious of whether or not it was the adenovirus. Anyway, we figured out that it had nothing to do with the adenovirus and had everything to do with whatever else was in there. Long story short, it turned out to be the discovery of Seneca Valley virus that came from either contaminated fetal bovine serum or swine trypsin. And that was how Seneca Valley virus and a whole new genus of the coronavirus family was discovered. So with that, I'd like to actually move forward and tell you some of the features about Seneca Valley virus. This is, I've spent 3 decades plus now working in the gene therapy field, as well as with oncolytic viruses and in cancer-specific agents. And this is certainly the most cancer-specific agent I have seen. And I'm going to give you some good examples here. So one thing we've done is we actually tested SVB by intravenous dosing at high doses in murine models, for example. And the nice thing about mice is that it turned out that mice were a very relevant model for Seneca Valley virus, and that's because murine tumor cells replicated SVB as efficiently as human tumor cells did. We didn't know the exact mechanism at the time, but that's for sure what we observed. So when you do high intravenous administrations at 10 to the 14 vector particles per kilogram, that's an enormous amount of SVB that was injected. What you can see here is you can see that with that immunohistochemistry staining there, you can only see SVB infection in the tumor, not in any normal organ. So we did not see any normal adult normal cells infected with SVB in multiple models and multiple murine models as well. You also see no microscopic pathology, same dose. You see very tumor-specific replication, and I already talked about the relevant model. There were actually several primate models that were done, and one was up to 10 to 11 vector particles per kilogram. Again, we saw no dose-limiting toxicity, and the same thing was actually done in porcine models, up to 3 times 10 to 11 vector particles per kilogram. Again, no dose-limiting toxicity and no infectious virus detected in tissue 7 days after dose, and that's the same with all the models. This was done by my previous company called Neoplasms. This was also done by the USDA in several occasions when Seneca Valley virus was first getting discovered. SVB has been also intravenously administered at very high doses in humans with minimal toxicity. SVB was tested in 3 separate clinical trials. There were 2 adult trials and 1 pediatric trial. And so there was 1 phase 1/2, which was an adult trial. There was a 1 phase 1/2, which was a pediatric trial, and there was 1 phase 2 trial, which was an adult trial. So there were a total of 76 patients that were treated with intravenously administered SVB. 49 of those patients were treated at 10 to 11 vector particles per kilogram. Especially when you consider the potency of SVB, we would consider that this is one of the highest levels of an oncolytic virus ever administered systemically. And we only saw 1 dose-limiting toxicity, and that was only because of tumor pain, which was successfully treated with an analgesic. So these are some of the results from the clinical trial. So we're going to highlight a type of neuroendocrine tumor patients called carcinoid patients. These were really the only patients that had 6-month survival, now considered to be a hallmark in the immuno-oncology field. And if you look here, we can tell if the tumor was infected by simply looking at the amount of virus in the bloodstream after a few days of the IV administration, it goes down to 0. And then if the tumor is actually making virus, you can detect it in the bloodstream. So if you look here, we can see anything above the red bar means that the tumors were infected. Anything below it, the tumors were not infected. And so you can see that these 6 patients had replication in the tumor. Interestingly, 5 of the 6 of these patients had a significant benefit, such as tumor shrinkage, as you can see here. You can also see symptom resolution. These patients tend to have what's called carcinoid syndrome, flushing, diarrhea, etc. The miserable type of syndrome. They had symptom resolution for several months here. Several of these patients also had marker declines. And so neuroendocrine tumors will actually make specific markers, such as synaptophysin A. And some of these patients had multiple benefits to the therapy. The same patient group, you could look at time to progression. And you can see the patients that had no infection had very fast time to progression. And 5 out of the 6 patients that had infection had very long time to progression, almost a tripling of mean progression-free survival here. We had some patients that had neuroendocrine carcinomas like small cell lung cancer, and of course the pediatric cancers. I mean, these patients didn't have, they only had the 3-month survival. We did have a patient that succumbed with small cell lung cancer. We were able to do an autopsy on that patient and get samples. And actually what you can observe here is that we see in the liver metastases of this patient replication, and we do not see replication of SVB in any other tissue, and this is in humans. This data has been published. We also tested a wide variety of primary normal human cell cultures, and this also showed no infection of SVB, and this data has also been published. So we didn't know early on for at least over a decade what the mechanism was. We had some ideas, we were testing it, attempting to try to figure out what the receptor was, but it wasn't really until CRISPR came about and our colleagues at Memorial Sloan Kettering used CRISPR to do a knockout library and figured out that the receptor for SVB was a protein called TEMB, or the gene name ANTXR1. That's what the receptor was. And what you're looking at here, this is published data too, is SVB complex with TEM8. And TEM8 binds in a crown-like arrangement around the spiral axis of the capsid here that you're seeing of SVB. And SVB binding triggers minimal changes in the capsid structure. So interestingly, TEM8, which is a Heretofore undruggable target, is selectively expressed in human solid cancers very similarly to the SVB selective replication in a variety of solid cancers and not normal cells. So TEM8 offers a very selective mechanism for any oncolytic virus, but Seneca Valley virus uses this receptor. So we are fortunate that that's the case. So TEM8's highly expressed in difficult to treat cancers, such as breast, colon, esophageal, pancreatic, ovarian, lung. And what we see here is immunohistochemistry of TEM8 on the surface of cancer cells, but what we don't see is any TEM8 expression on the surface of any of the normal cell controls here. We also see that it's expressed in multiple types of cells in the tumor, including malignant cells and cancer stem cells. These 2 types of cells, we know SVB infects these types of cells. TEM8 is also expressed on angiogenic endothelial cells, as that's how TEM8 was originally discovered by Brad St. Croix at the NCI. It's also expressed on cancer-associated fibroblasts, a very important type of cell to kill, as well as all of these. TEM8 is a complex integrin, molecules that are really notoriously difficult to drug. And again, no cancer therapeutic that targets TEM8 has advanced to clinical trials. TEMB8's also an adverse prognostic indicator. So where you see higher levels of TEMB8, you have much poorer, for instance, overall survival or percent survival as shown here. And it's been shown in every indication that's been examined. So TEMB8's involved in invasion, metastases, and it's just a really bad actor in cancer. So it's a really important cancer to go after. So given that, we took TEMB8 and we also developed, looked at 8 other genes, and we developed a 9-gene set. And this pretty accurately predicts whether or not a particular tumor, using a formalin-fixed paraffin-embedded slide, whether or not a particular patient's tumor will actually be able to replicate SVB. So this patient screening assay that we developed has a high degree of predictive accuracy, sensitivity, as well as specificity. It can be conducted in any CLIA laboratory. That was actually how the assay was actually developed to screen for this 9-gene set as well as TEMB8 expression. And when you do that, you can see which cancers actually are good targets for SVB. So here's the neuroendocrine carcinoma tumor, such as small cell lung cancer, a couple different types of breast cancer, both triple negative as well as ductal, prostate, sarcoma, glioblastoma. So these are really good targets, and this is a small list of the total. So shifting gears a little bit, all the previous trials were done with a single dose, intravenous dose of SVB as a monotherapy. And now there's a lot known about immunotherapeutics, the immuno-oncology revolution, and how to actually stimulate an immune response. So we went about doing a multi-dose regimen where we're doing intratumoral dosing of a type of cancer called PAN02 in a mouse model. So PAN02 is pancreatic cancer in this mouse model. It's a very cold tumor, and it's very difficult to treat. So we actually think we have some of the best data in this particular immune competent animal model. So in this study, we combined multiple dosing, intratumoral dosing of SVB in combination with anti-PD-1 and anti-CTLA-4, and these are the results. And so you can see, if you just look at percent survival in the survival curve here, that if you're treating with SVB or each one of the checkpoint inhibitors alone, all these animals succumb essentially by day 75. The anti-PD-1, anti-CTLA-4 control, these animals succumb by day 97. And when you have SVB plus both checkpoint inhibitors, essentially we get very long-term survival here, where these animals, starting right about here, are completely tumor-free. And none of these tumors actually never appear. And these animals remain tumor-free until the end of the study at days 165 when the study was ended. So very good eradication rate here. And then what we did is we wanted to see whether or not we had developed a systemic anti-tumor immune response. So this is how the study was done. So if you're looking at the primary tumor, so on day 41, we had some complete eradications on day 41. And so this is when we re-challenged the mice with fresh PAN02 cells on the contralateral tumor. And lo and behold, 83% of those mice actually rejected that re-challenge, whereas anti-PD-1 and anti-CTLA-4 group itself did not reject it. So when you have that triple combination of SVB and both checkpoint inhibitors, you get a very strong abscopal effect and everything that's indicative of a systemic anti-tumor immune response. So we also took tumors along the way, and we were looking for T cell infiltration into the tumor. And so what you can see here is this is the cold tumor. So you see very few other CD8 or CD3 cells within the tumor. This is immunohistochemistry for CD8 that you're looking at. With SPV, you're definitely starting to see some influx of T cells with, with the checkpoint inhibitors to some degree. But when you put all three together, you get a very strong influx of both CD3 and CD8 cells. That's shown in the immunohistochemistry here, and graphically here you can observe that. So I'd like to give you just an overview of the upcoming phase 1/2 clinical trial in neuroendocrine neoplasms. This includes neuroendocrine tumors as one group, and also neuroendocrine carcinomas, which include things like small cell and large cell cancers. So we've submitted the protocol to the FDA. We expect a response back from the FDA in the next few weeks. We've had many communications with the FDA in the past year or so. So it's one clinical protocol going from phase 1/2 into a randomized phase 2. We want to do the randomized because, again, TEMB is a very negative prognostic marker, and we want to be able to select patients for SPV infection. We already have 25 sites with experience in this area that are interested in participating in the trial, and so we hope to be able to get this trial running pretty quick. So the objectives of the trial for phase 1/2 is really to determine the safety and tolerability, and we'll be using that 9-gene set to pre-screen patients. And this will be done with SVB in combination with Ipi/Nivo. Then it will move into a randomized phase 2, and we'll be looking at progression-free survival, again, in combination with Ipi/Nivo in a 9-gene set with either neuroendocrine carcinomas or neuroendocrine tumors. So the phase 1 schema is a typical 3+3 escalation dose, and we'll be doing this with in combination with anti-CTLA-4 and anti-PD-1 or Nivo/Ipi. We'll start out single dosing. Here's cohort 1, 2, and then when we reach cohort 3 or the MTD, we'll then start backing off one dose. Then we'll actually start doing multiple dosing until we reach cohort 5. We would determine this to be the MTD or successful dosing, the one that we would want to move forward into the randomized trial. While this is moving forward, we'll also be doing a biopsy cohort of 6 patients at the maximum dose of multiple dosing of SVB, plus the checkpoint inhibitors, and closely examining the biopsy for a variety of markers. This is the randomized phase 2 schema. With neuroendocrine carcinoma, we'll be primarily looking at median progression-free survival. And with NET, we'll be primarily looking also at progression-free survival in NETs. Then moving forward, I did want to talk a little bit about our armed SVB program. So there's a specific place that we can put transgenes in that don't affect replication of SPV, so you still get that. And when you put the transgene in, what you end up finding is that the transgene is just as selectively expressed as SPV replication. And you've seen how profound that cancer-specific replication is. And we see the exact same thing with transgenes that we put in. So you can imagine the types of genes that we can put in here. Genes that will counteract the immunosuppressive environment in the tumors, tumor antigens, neoantigens, checkpoint inhibited genes, and genes that would actually kill some of those surrounding type stromal cells, such as cancer-associated fibroblast. And when you look at this in mice, when you do a high systemic administration of SVB-GFP, what you see again is you only see GFP in the tumor. This is a permissive tumor, and this data is published. But you see SVB, you see no green cells in any other organ. That's what's very important to really get behind. Again, this just agrees with how profound the tumor specificity is, and how you can also use this as a cancer gene therapy modality because you'll only get expression within the tumor. And these are the types of armed transgenes that we're actually putting in SVB. So you can see genes that are involved in immune activation, checkpoint inhibitor genes. You can imagine how terrific it would be if we could put in the anti-PD-1, anti-CTLA-4 and have that work every bit as good as the antibodies that were used in the study that you saw. And we also have some enzyme prodrug genes and some that are bispecific, including some that you might consider to be BiTEs. We also have a cancer vaccine program going, and we're in the midst of testing a model antigen called ovalbumin. So in summary, Seneca Therapeutics is really focused on developing its lead candidate to treat a variety of solid cancer indications, where SVB is extremely unique because of its profound cancer specificity. Again, this is partially due to the binding of the receptor, TEMB8. It also has a really interesting multimodal mechanism of action, stimulates an immune response against tumors. And we're planning on starting this phase 1/2 randomized clinical trial using SVB in combination with checkpoint inhibitors to treat patients with neuroendocrine neoplasms. We'll have the short phase 1/2 lead into that randomized trial. This trial we are anticipating starting in June. SVB is, for many reasons that I can't get into, an ideal candidate to develop cancer vaccines. We're developing multi-dose IV regimens, and we have a strong pipeline of armed SVBs. Thank you for your time, and please feel free to reach out to me with any questions.